So today we will have a seminar that is a little bit special because we want to focus on the question of open science and more specifically open data. And we have, so it's an event that has been organized, that was launched also by Noémie Cobolet a couple of months earlier, because there is a whole series of events which is called the Printemps de la Donnée. So there are many things in which you can participate. And just to remind the PhD students that you can sign the sheet here, it will be counted as transverse training for the École Doctorale. So we have the pleasure to host, unfortunately, remotely because of strikes. But we are very happy to have with us Lennart Martens, who is a full professor of systems biology at Ghent University, but also working at IPHC here on campus. He joined IPHC first with Cher Gutenberg, I think it was last year or 2 years ago. And so if you have questions for him, he will be here. He's often on campus, so feel free also to plan a meeting with him later. So Lennart will share his experience in the question of open science as a researcher. And then we will have a second part of the seminar where Jérôme Pansanel, who is just here, is a research engineer at IPHC in charge of the platform SING and also part of the ADELE Helpdesk organization. It will show you what are the tools available to you here to help you go towards that direction of open science and open data. So thank you, Lennart. The floor is yours for the seminar.
Okay, thank you very, very much for your kind introduction. And as mentioned, I should be back tomorrow on campus. So, and I'm in R2 in the buildings just opposite. So feel free to come by. Second office on the right. So yeah, unfortunately, I have to do this remotely due to strikes because I think the French like to make fun of Belgian people. I will help you a bit. So in Belgium, we have lots of strikes right now, and these typically tend to correlate with the formation of new governments. And so if you ever wonder why in Belgium we sometimes don't have a government for many months, it's because we like our trains to run. And so if we don't have a government, there's no reason to strike. But as soon as we have one, the strikes begin. So I wanted to talk a bit about the whole open science thing, because I think the open science thing is a very positive evolution of science. And I use this picture of Van Gogh to illustrate this in a way, because this picture is one of the early modernist paintings. And so the modern era was characterized by technological advances that allowed people to look at the world in new ways. And this created a lot of inspiration, both in the arts and in the sciences. And so a lot of optimism and positivism came from these developments. And this created opportunities. And these opportunities we still value today. Lots of developments in science that we built on, but also lots of art that we still enjoy. And I think open science is one of these moments that creates a new way to look at things and will create a lot of creativity. I hope to be able to convince you of that during my talk. So first, I will talk a little bit about why we should be happy to reuse other people's data. Some people say this is like toothbrushes, and you will never touch somebody else's toothbrush to brush your teeth, so you will also not use their data. But I don't think that is realistic. In fact, I think it's quite the opposite. I will then talk about 4 different types of data reuse that we can distinguish, because it helps you think about how you can use the data and how it becomes progressively more creative, but also a little bit more complex, but all doable. And so I will then illustrate these methods by 3 different ways in which we have used data in the proteomics context, which is in the life sciences, but it is chemistry adjacent because it's relying on mass spectrometry-based data of proteins that have been analyzed. So we're doing analytical chemistry in a way. We're just doing it in the life sciences. And so let's start by thinking, why should we reuse data? Well, in the life sciences for sure, but I'm convinced that the same happens in chemistry, is that a lot of the data that we acquire these days is what we call high content. And high content means that our machines, our instruments are so capable that when we apply them on the samples of interest, we obtain much more data than we actually need. And so a lot of this data is never used. This data just sits there and we don't talk about it in the papers, but it is there. And this is the bycatch. And it's not a problem, and it doesn't hurt us, but instead it is an opportunity. It is because this data can be used in other ways. And again, I will try and show you this. So very concretely in my field, in mass spectrometry, and the same thing goes with analyses of say, metabolites or small molecules in general in mass spectrometry, in chemistry, is that we acquire many more spectra. This is the blue line here. The total number of acquired spectra by the instrument is much larger than the number of spectra we identify. And of the spectra we identify, we only use a small portion in the papers because we typically don't care about all of the spectra. Many of them are trivial spectra from the point of view of the specific experiment we are trying to run. And so here in these identified spectra, there's a lot of spectra that are never actually interpreted downstream. They're identified and we know where they come from. We know what the analyte is, but it stops there because the analyte is of no interest in that particular study. And then, of course, there is also this gap between the identified and the total acquired spectra, which are very interesting because these are spectra we cannot assign to an analyte. And usually that is, in mass spectrometry at least, because we cannot imagine what that analyte might have been. It's an unknown, unexpected analyte. As a side note, it appears that this gap is widening. That's an artifact of the representation. In fact, this is pretty constant. So we tend to identify about 35% of the spectra. So that's high content. We have more data coming out of the instruments than we actually need. So there is a ton of data being generated that is currently not used. The second thing is that most of these approaches are also high throughput, and high throughput means you have a lot of data. And so you can see this in the repository that I started at EBI, which is now run by Dr. Juan Antonio Vizcaíno. And the joke is always that he started running the repository here, which is when it seriously took off. And you can see that there is a huge amount of data being deposited in this database every day, essentially. So in March 2021, for instance, we had 600 data sets coming in 1 month. That means if scientists work day and night, weekdays and weekends, that would be 30 data sets per day. And so there's a lot of data sets that run through these systems. And what is not shown here actually is that each data set tends to become bigger as time progresses. So a data set in 2013 would contain 10,000 spectra. A data set today contains 10 million spectra. And so in fact, you see there is a double exponential here. It is not shown, but there is just an enormous amount of data. And these two things, they really combine in a very powerful way. So there is a lot of the data, and very many elements in this data have never been thoroughly analyzed. That creates an enormous opportunity. Yeah. So in economy, they say you can only get something for free if previously you got nothing from that thing. And so here we get nothing out of a lot of this data because it's bycatch and there's so much of it. But that means that when we analyze it, we will get a lot of stuff for free. And so this is the idea behind all of this. So we do experiments. These days, in most fields, we have to do some kind of computational or statistical or mathematical analysis of the results to interpret them. And then once we have the results, we need a PI to create a Nature paper. And this is how the flow of science works traditionally. But if you have lots and lots and lots of these people who then all submit their data into central repositories where it can be used and reused by other people, well, you could just tap into that vast amount of knowledge and create novel research. I usually target this slide to young people. So you can see, I hope, where my propaganda element is in this for young people. I'll give you a moment to locate the propaganda element. And it is, of course, the absence of the PI here. So you don't need these old PIs anymore to help you get Nature papers. You can do everything yourself. But fundamentally, this all stupid jokes aside, this is a very real possibility. And I will show you many examples of this as we go along. A little bit more formally, what does this look like in an open data exchange ecosystem? That there is an experiment which leads to data that is interpreted. This interpretation then yields new knowledge. This new knowledge can be disseminated traditionally in papers or in patents, for instance. You disseminate your knowledge. And then this knowledge is then built upon by other people because it inspires new experiments and the cycle continues. Now, if this dissemination is done well and the data is reusable, as open science tries to ensure for data sets, then this can be shortcut. And as I said, because we have high content and high throughput data, we can then start interpreting this high volume of as yet not completely used data for novel interpretations without having to pass through experiments. And so in a way, if you want to convince the funders of this, it's very easy. You tell the funders, if you perform open science style experiments, then the value of the generated information is augmented quite substantially, because now it's not just a single group that analyzes this data. No, there are multiple groups who can also analyze this data in their own way and do new things with this data. And so the 4 types of data use that I will talk about is straightforward use, where you would just take the data and do more or less what it was intended for. You can reuse it, which means that you can take the results and use these in a different way. Then you can reprocess, which means that you're going to look at the data in a completely novel way. And then finally, there is repurpose, where you actually say, no, all of this original ideas about the data are fine and well and good, but I have this completely crazy idea in which I can use this data or large ensembles of data in ways that nobody ever imagined. And I'm going to show you an example of reuse, of reprocessing, and of repurposing that we have done on proteomics data, which I then hope will inspire you to think about possibilities in your own domains. So these 4 types of data reuse, in the identification of proteins, I can give you some examples. So a straightforward use is the fact that once you identify a protein, you can then log that in a database called UniProt, which keeps track of everything we know about every protein in, say, the human proteome. And so you can go to a protein, say, beta-actin, which is a skeletal protein that keeps all of our cells in the right shape. And then you will find a lot of information that we know about this protein based on all of the identifications and all of the context that we have generated in the literature. So this essentially is like a catalog of proteins. It's very much like a Sigma catalog of a chemical compound, where you read everything we know about this chemical compound. Or it could be like, say, a classical toxicology lexicon, where you can read all of the toxicological information, say, about the chemical. And so that's straightforward use. You just aggregate all the knowledge and put it in one place. That is supremely useful. We all use this all the time, right? So this is kind of a no-brainer. A second thing that you can do is reuse, but I will give you another example, and that would be based on the fact that we have identified proteins, we know how it behaves as an analytical entity, and we can use that knowledge to start making targeted approaches. And so targeted approaches allow us to go and hunt for that analyte very, very specifically, because we know the kind of footprint it leaves in the data. And so this was done in the SRM Atlas, which is something where Christine Carapito of the IPHC during her postdoc years made seminal contributions as well in the field. And so that is another type of catalog, but it's a catalog that attempts to extract the information at a slightly more sophisticated level. It's not just aggregating, it's interpreting the information during the aggregation. And that is supremely reusable information. And then as it comes to reprocessing and repurpose, I won't talk too much about that right now. I will show you the examples. I think that will be much more clear. In general, there is a trend by some people to say that open science is there so we can police other people, so that we can look at their data and say whether it was done well or not well. That happens. In particular, in analytics, people are very happy to say that data was analyzed in a poor way. But to be quite honest, this bad cop approach, I'm not a big fan of that. The reason for this is that we already can police everybody else without data being open. There are many examples of fraudulent papers having been found out without the source data being shared. Plus, if somebody really wants to commit fraud, they will also tamper with the source data. And that is not always obvious to detect. And so fundamentally, this is a really bad reason to do open science. We don't need it for that purpose. But I think there is a much more positive trend to this, which is when you do a fantastic experiment, then people are very interested in having a look at your data because it will enrich the field, it will enrich their possibilities and their opportunities. And so people are very, very happy to use and reuse your data. Just like you probably should be very happy to use and reuse other people's data. Again, they're not toothbrushes. And then finally, I put this in gray so because I don't want to offend most people, but there is a fundamental truth emerging that is that once data is being shared, your data sets tend to live a much longer and more useful life than the publication associated with the data set. Publications get out of novelty very quickly, and they're very quickly superseded by better publications. This is because science advances very quickly. It could be your own publications that supersede your old publications. It could be a competitor or a colleague that does something that adds to what you have done, and so that becomes the new standard. But the data set that you generated, that will remain valuable. And in fact, it might even become more valuable as we understand more about where the data came from, both the sample and the instrumentations, for instance. And so this is a key thing to realize, is that the ultimate product of our science is probably the data. And the paper is a byproduct of the data. It is your byproduct, and it's very personal and it's very nice, but the data is actually the fundamental product. And it will probably be the most useful over the long term. Okay, so enough about the background stuff. Let's see how we can put this in practice. So the first thing that we did was a reuse of available data. And with reuse, what we did is we took a lot of data where it says, this is the analytical signal, and this is the entity associated with that signal. So it's a spectrum and its identification in mass spectrometry terms. And what we have done is we have tried to build machine learning models that take the identification and predict the analytical signal, so that from the identity of the entity, we get the mass spectrum. I will make this long story very short. So we started this work in 2009, first published it in 2013, and then kept updating it over the past 12 years. And it still is one of the key tools in the field. It's called MS2PIP, and this is a real spectrum. So it's a mass spectrum, masses and intensities. This is the sequence of the peptide, and so it's a series of amino acids in a linear chain. And when you fragment this in a mass spectrometer, this is the spectrum you get, the real one. And this is the prediction from the tool. If we focus on this area here, because it's nice and simple, you will see that actually the prediction is very, very good. And what we have shown, meanwhile, in the field in general, not just us, but other people with us who have also taken different approaches to solving the same problem, is that we have reached prediction accuracies that are equivalent to the reproducibility of the fragmentation. So we have reached the theoretical limit. We can predict these spectra as good as our machines can record them, which is really, really great, because that means that we now know exactly how an analyte behaves in the instrumentation, which also means that we can make the prediction and then correlate what the prediction looks like with the experimental result and say whether we have found a match or not. We have not only done that for the mass spectrometry element. In mass spectrometers in the life sciences are fed by liquid chromatography systems. And so these liquid chromatography systems deliver a physicochemical property of the peptide, which is its retention time on the column. In our case, it's a reversed phase column. So we have a stationary phase that is hydrophobic, and we have a mobile phase that is a mixture of hydrophilic, hydrophobic, and it becomes more hydrophobic over time, thus eluting progressively more hydrophobic peptides from the column. And what we can do is we can predict these retention times. So we can say, given this sequence, this peptide will elute at this point. And we cannot just do this for the peptide sequence. We can also do it for the peptide sequence carrying particular modifications. In this case, there's a variety of acylations. So acetyl 2 carbon groups, succinyl 3 carbon groups, and propionylation with 4 carbon groups. They get progressively more hydrophobic, and we can actually predict the effect of that. So with traditional prediction, which does not take the modification into account, would be offset, because we underestimate the hydrophobicity because we don't take into account the acyl group. But as the acyl group is taken into account, we can reposition that thing. Very interestingly, and something that was unexpected to us, was that the machine learning algorithm actually also increases not just the accuracy by placing the red line back onto the dotted line, which is where it should be. That's an accuracy shift. It also increased the precision. So the width of the distribution in red is bigger than the width of the distribution in blue. And that is shown in these box plots, where you can see that the blue box plot is always narrower than the red box plot. So we get precision as well as accuracy. And we use these things in the interpretation of this data that comes off the instrument. So as we get data from the instrument, we get scores for them, and we say, so there's traditional algorithms that say this peptide sequence is the most likely originator of this spectrum. But now we can say, well, actually, if this is the peptide sequence, we expect the following spectrum, we expect the following retention time. So let's see how well these two correlate with the actual measurements. And we can do a much more detailed and much more fine-grained analysis of this. And this allows us to separate true positives in blue much better from false positives in red, fundamentally allowing us to identify more of the spectra. And so what actually happens, and this is important to realize, is that people have shared their proteomics data. As a result, we had millions upon millions of data points to train very sophisticated machine learning algorithms, of which I have just shown you that the performance becomes really good because we have so much data. And then we will use this, this information, this new prediction algorithms to improve the identification of the mass spectra. What happens is these tools are also open source and freely available to anybody in the field. Thus, everybody who contributed the data originally, thanks to the fact that their data led to the development of these tools, can now use these tools and can improve quite dramatically the results that they obtain from their analyses. And so this is a very clear virtuous cycle where the delivery of the data very quickly, this is in the space of a few years, led to a major improvement in our overall analytical capability, thanks to the creation of better software. So it really, I think, showcases very nicely how well this can benefit a community in general. Now, this is all open source, and I mentioned that we do open science. All these tools we built are open science, but that does not mean that this is incompatible with commercialization. I just wanted to point that out. And so the commercial companies like Bruker, whom you probably know if you're in chemistry, and Matrix Science, who are more specific to the proteomics world. So Bruker, of course, is an instrument vendor, whereas Matrix Science is a software vendor. They actually use our tools, MS2PIP, DeepLC, IM2Deep, and here MS2PIP, but they have DeepLC as well, in their software. And we work with them. And these are commercial pieces of software that incorporate our software. And so all you need to do is do the right kind of licensing. So I don't have a lot of time to talk about this right here, but if anybody's ever interested in this, feel free to come and talk to me about this, because I have some experience in how you handle these kind of things. And it's not as difficult as you would think to do something in open science that is nevertheless compatible with the exploration of some form of commercial enterprise. And what is interesting, of course, is that you can see the benefits of using our tools, which yield 13, 20, 18% better identifications, or here in some cases, you boost by 60% or by 54% the identifications. Fundamentally, the difference between these 21% and these 54% is the 21% is for very standard data, where we were already very good in the analytics. These higher numbers at 50, 60%, they're typical for analyses that are far more complex and far more difficult. So what actually we learned from doing this is that the machine learning tools that we built based on all the analytical data are particularly useful for the most challenging types of analysis. And I think it's not too much to state that thanks to these machine learning algorithms, people in the field of proteomics are now capable of doing analysis that 10 years ago were unthinkable. And so this really, really lifts that community up, and is all based on data sharing. There's a very clear one-to-one relationship between these two things. One example, this is with the University Hospital here in Strasbourg. This is with Professor Siamak Baram and Professor Rafael Carapito at the hospital. And they were looking at patients with cancer, and they want to find neoepitopes. What are neoepitopes? These are small bits of the cancer cells that are unique to the cancer cells, and that can be used to train the immune system of the patient to go and attack the cells that have these elements. So essentially, we're teaching the immune system, this is what a cancer cell looks like, now go and kill it. And it's the most promising type of cancer therapy that we have today. But it's complex and it's early stage. And the key thing is find the unique elements. And to find the unique elements, you want to find as many as possible elements that are presented on these cancer cells. And here's what the effect of our software is on this. So if you don't use the software, you have about 2,000 of these elements that you can see presented by a cancer cell. Most of these are not unique to the cancer cell. If you use our software, you end up with 3,433. So effectively, you're adding 50% or more to the total count of elements that you find. This is a filtered set where there's a particular filter applied, but the ratio holds. Yeah. Now, when you see this, it's a lot. It's a dramatic increase. It's like I said before, it's this 50-60% increase, and so you can become suspicious and say this cannot be correct. It cannot. This is too nice to be true. And so what they did in the hospital is they did not trust us, and so they made a plot of the HLA typing. This is essentially your antigenic profile. And everybody of us has a unique one of these, which is why it's very difficult for me to give you a kidney. We need to be compatible in these HLA genes, otherwise your immune system will reject my tissues. And so the compatibility between these things is what makes people possible donors for other people. And so since we all have a kind of a unique signature, this patient in these fragments showed this distribution of different HLA types. And when we look at our results, we get essentially the same distribution, meaning that our results are meaningful. Because if we add 50% random nonsense, this would become very different, but it doesn't differ at all. So we find the same HLA typing. And this is at the point where then the medical people say, so your approach seems to work because we find very relevant data. And our approach obviously does not know anything about HLA types. So it's completely blind to that. And so here you see an immediate application again of data sharing leads to better tools lead to applications right here, right now in translational science. To talk a little bit about chemistry, so we have predictions of retention times for small molecules as well. And there's a lot of tools built for this. The problem is very few people use these. And the reason why few people use these is because there are so many different ways in which you can do chromatography on small molecules. And if you then correlate one way with another way, randomly selected samples, you get these very nonlinear relationships. So the retention time in one system bears very little relationship to a retention time in another system. And so the way that people try to fix that is by calibrating and so forth, but calibrating tends to be linear and tends to be very difficult and tends to be setup specific. Plus, calibration relies on an overlap in the actual analytes, which may not always be present. So here we did something similar. We used a lot of data where we actually train based on a given setup. We train a model that predicts retention times, and then we apply that to another model. And obviously the predictions are not very good because this other data set comes from a completely different system and the predictions are bad. Well, maybe they are bad. So that's what we evaluate. We evaluate the predictions that we get from the wrong model as applied to this data set with the actual values of the data set. And sometimes you see this very bad nonlinear behavior, which means that this in fact is a very bad model to predict this setup. But sometimes you're lucky and sometimes you get a good version. And so this is what we do. We take a lot of data sets. Again, they come from the public domain because they are shared. We built multiple machine learning algorithms because it depends on the style of machine learning algorithm, which is the best. So we produce multiple so that we can always pick the best performer amongst the multiple. And then we have a setup specific model. And then that is being applied to a given data set that we don't know yet, and we apply each model to this data set. We calibrate the model by comparing the real retention times with the predicted ones, as I've just shown you, and then there is a few possibilities. Imagine that on data set 1, the model actually performs really well, because this model is nearly linear with the original data set. And so that would then be a very important predictor in our total predictor, because we're going to aggregate all of these. Now, on set 2, however, we get a very poor predictor. And so that will be downweighted on this particular data set, because for this particular data set, that particular model is not good at all. But nevertheless, there may be some compounds in this data set 2 that are simply not represented in data set 1, but they are present in our set X. So in the land of the blind, one eye is king. And so if we don't find any element in set 1, well, we will nevertheless choose the prediction from set 2. It is a worse prediction, but it is still a prediction and it's better than nothing. And so that's why it's still included. It's just downweighted. It becomes the option of last resort. Now, we call this thing whimsically call LC based on calibrate all LCs. And what we effectively do is that we take everything that everybody has ever done, and then we apply all of that expert knowledge to your particular data set. And so we can actually learn from all of the existing data set to make the interpretation of your data set better. So what are the effects? Well, the effects are that when you set up specific models, your performance here measured as the average error that you make on the prediction is pretty poor. Especially if you don't have a lot of training analytes. When you have 20, it's much worse than if you have 100, because the more training data you have, the better the model becomes. If you use calibration, classical calibration, linear calibration, you see that you do better than just building a model, because you take a model and then you adapt it to the setup, which is always better. But the best thing is when you use all data sets. And that's what you see in these blue bars here. And you approach that progressively, but you always gain compared to calibration or compared to setup specific models. So no matter how many analytes you have, this remains the better option. It also tends to have lower variance, so better precision. And this is what you see here on another model that tries to adapt the different circumstances, but that does not take into account all of the public data. And it does a reasonable job. It does a good prediction. It would be competitive with calibration without a problem, but it's not competitive with our approach. Because our approach leverages strength, as statisticians would say, across all of the public data. Now, what can you do with this? You can use it to identify, for instance, lipids. And here you can see how that predicted retention time correlates with precursor mass as total information to identify, say, either a very specific lipid molecule, which is very difficult, or a subspecies where, for instance, we don't exactly know how many double bonds there are, or maybe where the double bonds are. We can take a specific species of lipids, or we can take the main class of the lipid. And what you can see is that the precursor mass alone becomes progressively more useful as the information you obtain becomes progressively less useful, because it essentially in the end is telling you it's a lipid. So that's not really that informative. But that at the higher informative stages, which are essentially these two and possibly the species, that the combination of the precursor mass with the predicted retention time, which is the red line, the red bar here, that becomes the most sensitive. And so again, for lipid identification, there is already a clear advantage. And the percent of identifications goes from 20 to 40, so we're doubling. This is 2 times as good if only you just predict the retention time. And why can we predict the retention time in a way that works for you? Well, because we have tons of data and we have a clever way of adapting those tons of data to your situation. So again, it's the same story as we had with the proteomics, just applied to chemistry, same logic and a little bit of creativity. Something that we do with this for companies is companies, especially pharmaceutical companies in the quality control of drugs, they want to find out whether there are contaminants. And so they have a list of possible contaminants and they do quality control checks on all batches to see whether these contaminants are present or not. And if they're present, they pull the drug quickly and they don't ship it. And so they want to know what is the optimal chromatographic setup to separate clearly all of the contaminants that I may expect in my production batch. And so that particular question can be answered by these algorithms as well. And so you can see a very clear translational implementation in business in a place where it makes a lot of difference. And so this you're talking about millions of dollars here. Each time such a batch would go out and would create problems. So purely based on the predictions, you can tell them in advance what is the optimal gradient. And so how does this impact the company's bottom line? Well, very simply, if they have to do this manually, it will take them 3-4 months. These 3-4 months are 3 or 4 months during which they cannot ship the drug yet. And if they cannot ship the drug, the patent is expiring. And every month that the patent is not yielding them return on investment is a problem for the company, because we're talking, again, about millions of dollars or euros, if you want. And so this pipeline allows them to check the collective memory of all the chromatography that they have ever done in-house. And it tells them that particular chromatography is highly suited to this problem. And it's fun because they can use the stuff that they have done in the past. So they already have the protocols, they already have the instruments, they already have the experience. And what is particularly relevant is that ultimately they choose the setup that they want to use. The algorithm gives them options, but the humans decide, which is a very important factor in getting AI accepted, but that's a different discussion. And what is fun to notice actually for everybody involved was that, because we have done this with actual pharmaceutical companies and they're using this in production, is that very different kinds of chromatography are selected. And the humans usually say, this one I also had in mind, but the other one I would have never thought of. But if it so happens that today in their current production, this is a far more commonly used chromatographic setup, it makes much more sense to use this system than it would to use this system. Even if the performance of this is 5% lower, it means you don't have to set up a whole bank of this system just to track the quality of one product. It's much more easy if you can keep the same system running that you already have. And so this is, again, very, very easily translatable into real life application with commercial implications. Maybe if you're thinking about, can I use other people's data in AI models, this very complex slide, which you should not look at in too much detail, essentially proves that both in the US and in the EU, if you train an algorithm on data, on other people's data, if it is in the public domain at least, there is no copyright that is transferred. So fundamentally, you can just use this. Right. So that is reuse. So we use the data relatively straightforwardly. I'm now going to go a bit faster, but I'm going to show you some reprocessing results. Remember that with our tools, we can go deeper in the data than we could go before. And so one of the ways in which we can do that is we can look for these unknown analytes. Remember that we had the spectra identified and the ones that were not identified, and there was this gap. And so now what we're going to do is we're going to find out if we can learn something about those unidentified analytes, which are very interesting because we don't identify them because we don't know them yet. And one of the ways in which we can identify them is through scanning for post-translational modifications or chemical modifications on the proteins. And so you see that here blue means unmodified and orange means modified. And what we see is that we find lots of modified proteins. In fact, proteins are modified to the hilt. And so we then said, if we can go back to old data and look at the unexplored missing data, kind of the spectra that we could not identify, if we do this at a large enough scale, we will get a landscape of protein modification across a lot of different tissues and proteins. And so we did that for mouse and for human data. We took the public domain data, which is about 600 million spectra for mouse and 1 billion spectra from humans. And by the way, just to give you scale, this is 6% of the publicly available data. So we only took the tip of the iceberg because we did not at the time have the compute power to run everything. We're now trying to run everything. For humans, we only have 4% of the available data. But that already gives us 2.9 million modified sites in mouse and 5 million modified sites in human. Nobody has ever seen this. Well, in fact, everybody has always seen this, but they could not interpret the data. But because the data is public, we could build the tools to go back to the public data and extract this information from the experiments that were done in the past. And so this is what it looks like for a random protein. This is ATP-dependent RNA helicase. It's a very typical protein. It's kind of abundant. It's nothing special. And so these are all the modifications on the protein sequence. And there's lots and lots of modifications. Interestingly, there's lots of chemical derivatives. One of the things we learned from this is that there's a lot of chemistry happening in a cell, actually fundamental chemistry. And so the cell is kind of leaky when it comes to that. And proteins are constantly exposed to chemical modification due to random chemical haphazard effects in the living cell. And if you look at, say, the biological ones, these are the post-translational modifications. One thing that we see is that they're all on the outside of the protein, which makes sense. That's where you can access the protein to modify it. And that practically the whole surface area can, in principle, be modified at one point or another. And so this is teaching us a fundamental lesson about proteins, i.e. proteins are not naked molecules. Proteins are heavily modifiable and are typically heavily modified in vivo. And these modifications affect their performance. This is a very fundamental new way of looking at proteins. And this is because we can reprocess the data. We can look at the data in new light and find unexpected signals in this data, which open up our perspectives of what is actually going on in a living cell. Interestingly, we can find disease markers, and these are both interesting for chemists, I think, because we find chemical artifacts, if you like. So kynurenine, for instance, is heavily reduced in Alzheimer's disease patients. So proteins have a lot less kynurenine. Now, I didn't even know what that was, so I had to look it up. And kynurenine is in fact a thing that a modification of tyrosine, of tryptophan, sorry, that is involved in the creation of serotonin. So it's an oxidation set of oxidation reactions. And the thing is, the bioavailability of serotonin and kynurenine is very much reduced in the urine and serum of Alzheimer's disease. And we see it on the proteins. So the proteins contain less of this chemical effect. And so these can literally be used as a biomarker, because you see on the surface of the proteins the physiology of the cells changing. This is an opposite example where something in disease, in this case, older people is much more prevalent than in young people. And this is methylglyoxal. And methylglyoxal is an advanced oxidation end product of glucose. And so what happens is in aging, your glucose levels are not as well maintained, homeostasis maintained as in young people, and you have a lot more oxidation going on, and therefore oxidative damage, and therefore also oxidative degradation of your key metabolites. And so your glucose is being oxidized much more than in young people, and these oxidation products of glucose then stick to your proteins. So I call this barbecued proteins. And it's like the black stuff that carbonizes on your barbecue. This is kind of what is happening in your proteins. Your proteins are starting to look like a well-used barbecue as you age. Okay, then final thing, repurposing. So reprocessing is cool because we go back to the data and we look for things that people have missed. And then we find lots of these things. And these are all interesting because people tend to miss signals because they cannot yet interpret them because they are unknown, unexpected. And when you find unexpected things, you have breakthroughs. Now, repurposing, we're going to do crazy stuff that nobody ever thought about. So this thing of identifying stuff in the data, that's what everybody wants to do. But what we're going to do now is completely crazy. So the first thing is we do a very silly thing. We say a proteome is a set of proteins, and proteins are tools for the cell. And so if cells have different functions, they will have different tools. It's like a carpenter has a different set of tools than an electrician. And that is in fact true. So if you use the proteome, just the list of identified proteins, you can predict the tissue type, whether it's eye or heart or kidney or liver. You can predict that very, very accurately, which is logical. And we can even do that for types of cells. Occasionally, it doesn't work so well if we don't have enough data. So that's why a few of these are off, because we simply don't have the data. But when we have data, we can predict perfectly accurately, essentially, the tissue or cell type of origin of a data set. Now, this seems backwards because, people have a sample, they know what the sample is. So why would we have to predict where it came from? We already know where it came from. But the interesting thing is we trained the system on healthy tissues. Now we give it a diseased tissue. So it has never seen a diseased tissue, but we give it, for instance, lung cancer here. If you give it lung cancer, the system will say, this looks like lung with a 53% probability, which is very bad, by the way. You can see here the prediction accuracies are about 90 to 100%. That's why they're dark blue. So 50%, it practically never happens. So this is what 50% looks like here. So the system says, this kind of looks like lung, but I'm not convinced. And then we can use explainable AI, where we ask the algorithm, tell me why you think this is lung. And these are these proteins in red. And so these are fibroblast specific or enriched in lung, and we find them. And this one has low to no expression in lung. And what is the amount we have found? Nothing. So the absence of this protein is taken by the system to mean that it is lung. So the absence of this protein is a lung marker. So it can take absence or presence and use both of these in its determination. And we can actually see that this correlates with prior knowledge. If we look, however, on the things that make it say, this is probably not lung, these are in blue. So this is all the stuff that should not be in lung, but somehow is there. And what you can see is that these fibroblast things are now gone. So there are proteins that are missing that should be in lung, but they're gone. And on the other hand, you see two proteins that are there that should not be there. And this one has been known to be observed in lung cancer, and this one has been known to be found in lung obstruction and lung cancer. And so this system is at the molecular level classifying tissues, diseased tissues, while telling us these things make it still lung, these things make it cancerous. And so this correlates extremely well with what we already know, indicating that we can use systems like this to do a complete analysis of diseased tissues. And in fact, it works not just on cancer. This is celiac disease. Celiac disease is an inflammatory bowel disease. So it's completely inflammation. And what you can see is that system predicts small intestine. I did not put the probability. The probability is 11%, so it's very low. And it says this is why it's intestine and this is why it's not intestine. And if you go and look at it, the red ones are small intestine specific and gastrointestinal epithelium. So again, known markers. And when you look at the things that say this is not the small intestine, you will see that everything is immune related. And so this is logical because this is an immune-related disease. This is an inflammation disease. And so, interestingly, one of these is a known celiac disease marker. Now, this study was actually done in a quantitative way. So it was an actual differential analysis study where they found that indeed this one was significantly downregulated. And so meaning that, you know, this small intestine likeness is being reduced. This one was not found regulated, but we found it anyway. And then you can see that they found two of our markers as significantly upregulated. So we agree on those, but the one known disease marker was missed in the quantitative analysis because it was below the threshold, but we found it. And so this is clearly helping in a way that is synergistic with classical studies. And keep in mind that this one is not just trying to differentiate between healthy and diseased versions of small intestine. No, it's looking at the diseased small intestine. It's comparing it to all the tissues that have ever been seen. So we're not doing a simple experiment. We're doing a much more complex experiment against the compound background of all tissues and their identifying features. And so this, again, is super powerful. We had not even expected it to be this powerful and this interesting. I also mentioned that we have modifications on proteins. We're also using modifications now as input to this algorithm, which is making it even more powerful. And so this is one of the examples of repurposing. We do something completely crazy that nobody who was making these original data sets would have ever thought somebody would do with their data. And the only thing it took was one, having the data, and two, having a little bit of creative ideas. The rest of this is not that difficult. I had a student from the ECPM do his thesis on this work. He did a fantastic job. He could replicate and reproduce this entire study in 2 days with a bit of help from ChatGPT. He could generate the code in 2 days to do this work. So it's not complicated. It's just, it requires a little bit of ingenuity. And then the final thing I will talk about is another repurposing, just to show you how crazy you can get. And what we do here is, it's complex. It uses variational autoencoders, but the bottom line is we look at guilt by association. So we're essentially saying we have lots of different experiments. If two proteins are always shown together in these experiments, that probably means they have something to do with each other. And it turns out that what they have to do, that this works, and that they actually are linked through their function. So they are functionally linked. And what we can do then is we can make these big maps of proteins that link with each other. A colleague of mine from Leuven University once asked me, Jos Chimkovic, he said, he's into chaperones. Chaperones are proteins that fold other proteins correctly. And he said there's about 300 known chaperones, and they all have their own clientele. They all have their proteins that they fold. And so he said, if you have such a fantastic approach to do this kind of stuff, show me the list of clients for each of my 300 chaperones. And so we did that. So Michaela Cutrulli here, she actually went into the system and got this data out in 5 minutes. And then one of Jos's postdoc made the map, and then the squares are all the chaperones, and you can see all the client proteins for all the chaperones, where there is some overlap, but there are also chaperones who have clear, unique proteins associated with them. And so these kind of maps come from a table that is essentially a bunch of experiments and a bunch of observed proteins. That's it. Okay, this is clear biology that's coming out. Even more interesting, we have modifications. What you see here is you put end-to-end all the protein sequences of all the proteins in the human genome, and then you annotate all of the phosphosites. So these are all the phosphosites on all the human proteins that we've ever seen, the complete human proteome. And we correlate that with itself. That's why the central line is blue. That means every phosphosite correlates with itself. And when it's phosphorylated, it's phosphorylated. But when we zoom in, I'm going to zoom in on this section here. When we zoom in here, you see that you have the small square, blue square, and it's a dark blue and a light blue square. Now, these are on a single protein coordinated phosphosites. That means that multiple sites on this protein are phosphorylated together. Even more interestingly, since part of it is light blue and part of it is dark blue is always there. Light blue is sometimes there. So dark blue is the driver. Light blue are the passengers. The dark blue one has to happen for the light blue ones to come along. And so that information is just coming from this data set. And of course, that's at the micro level. If we look at the macro level, which I find more interesting, you see these large patterns of phosphorylated proteins. And you can see which proteins all phosphorylate together. And you can see and you can also study in which context, in which organ, in which tissue, in which cell type, under which conditions of health or disease do these things happen. And what you can do is you can then extend that even further to different kinds of modifications. So here's phosphorylation and methylation and acetylation together. And you get these large clusters of proteins that all modify at the same time. Sometimes they're mixes, like here phosphorylation and methylation. Here it's nearly exclusively methylation. Here it's nearly exclusively acetylation. Here it is a mix of all kinds of things. And all of these clusters can then be analyzed for their biological roles. And it turns out that many of these have very specific biological roles. This is amino acid metabolism. Then here is a giant cluster of methylation that is immune system related. And then we have other clusters that are, for instance, related to, that was phosphorylation. The methylation here, this is the immune related one, I'm sorry. And this one is phosphorylation, and that one is related to cell signaling. And then we have one that is only tyrosine phosphorylation, that is here at the bottom. And that one is actually related to cell migration and cell mobility. So clear clusters with clear biological roles that all coordinate their responses. And all of this, again, comes from the same table, a bunch of experiments, a bunch of observations. And again, all it took was a bit of imagination. Again, this is not that difficult to set up. It's not rocket science. Anyway, with that, remember that I talked about a new way of looking at things and how open data, massive amounts of data can help you look at new things. I decided to create a Kandinsky, which is also one of these modern arts, based on Joost Chimkovic's network. So I took this network and I asked an online generator to create a Kandinsky based on that. And that's this one. It's something I would probably hang in my living room, but I am very much afraid that Joost would call copyright and make me pay for it. So I'll just keep it with on the slide. And with that, I thank you very, very much for your attention. I'm super happy to take any questions. And do keep in mind from tomorrow, if the strike is over, but it should be, tomorrow I should be back in Strasbourg. You can always come by and talk to me about things if you want. Thank you very much.
Thank you, Lennart, for the very nice talk. For the questions, we need you to ask the questions in the mic, so please raise your hand with the mic.
So Noémie Covolet, I'm in charge of open science. Thanks a lot for your presentation. I have a question about having data. How do you select your sources? I saw in your presentation that you mentioned the PRI database and databases that are giving access to data that are under public domain. And also you mentioned some data sets from preprints sharing bioarchive platform. But when you need massive data sets, how do you select your repositories? And are the metadata enough? Or do you need to have contact with the producers? How do you do with the part of the before tracement? How do you manage this step?
Yeah, so I actually have slides on the second part because I didn't have time. I already talked far too long, but I have a few slides. I can show you a few interesting things about metadata, which is the biggest problem actually. Data is much easier than metadata. But I'll first talk about the repositories. Where do we find the data? Well, in Pride, because I built Pride. So this is what I did as a PhD student. I built a database to hold the information because I wanted to do the stuff that we're doing now. For those of you who are interested in knowing the timelines, I started my PhD in 2002. So it took me more than 20 years to get to the stage where we can actually do this kind of work. It's not easy, but the only way in which we could get enough data was to build a centralized repository where everybody sends their data to one place. Now, of course, that never works, because when you have one in Europe, they want one in the US. And when they see that they have one in the US, the Japanese want one, and then the Chinese want one. And so everybody builds their own repository. So then the next thing you need to do is build an international consortium of people exchanging data between them. And that is Proteome Exchange. You may have seen that. I'll quickly go back. And so this is the PRIDE database, but usually I actually correctly call it the PRIDE Proteome Exchange. There we go. So the PRIDE database, which is the key, the center point, the hub of Proteome Exchange, which is the global network of data exchange. So this is non-trivial. So you have to set up means, an ecosystem in which this data can be shared. I also pointed that out in the beginning. I'm sorry for epilepsy-inducing flashing of slides here, but that is essentially this point. It doesn't say data repositories, and it doesn't have a direct link from the papers. You need to have the central infrastructure, and building such central infrastructure within fields is essential to create this open data exchange ecosystem. So I will not trivialize that. That is a lot of hard work. Having said that, there are people doing this. And I'm sure in chemistry, there are more and more of these resources right now. I already know of a few based on for instance, the mass spectrometry world increasingly has these kind of things, but that is critical. So that is fundamental infrastructure. Otherwise, you cannot do it. Second thing is about metadata. So getting data, we started doing this. I started PRIDE in 2005. And so the growth curve, as you can see, when did it become usable, actually, this data in PRIDE? We started doing predictions here in 2009. That's when we had enough data. By the way, this is not a cumulative chart. This is the amount of data sets coming in every month. So you have to imagine the cumulative one by summing all of these things together. But cumulative, it's a hockey stick. It looks like this. So by 2009, end of 2009, we had enough data to start it. Today, we have much more data. We have much more data. So the algorithms only become better. And then that's fine. That's sorted. But metadata. So if I go behind my slide here, this is the metadata. This is originally published in 2015. So you can see actually this was 10 years of proteomics public data. I published this paper to make a point. These are the different mass spectrometers that people use. And you can see that the most popular mass spectrometer is unknown. So people refuse to just tell us the mass spectrometer. This is a data set of mass spectrometry data, and people don't tell us the name of the instrument. So at that level, it was that bad, actually. And so we tried to fix that. We first tried to fix it with better software for submission. And then it turns out people don't use that. So it doesn't make a difference at all. So the fact that it was easy to annotate did not help. The thing that helped, because you can see it declines at some point, the unknown starts to decline and the knowns start to jump up. Yeah? And this was the most popular instrument at the time. And you can see that because that's the one that jumps. Why did this work? Because we put a policeman. And so by that time, submission to the database, to the repository, was mandatory for publication. So we got to that stage. That's a fight in its own. Again, if people are interested, I can have very long talks with you about this, about how to do it and how not to do it. But we succeeded. And since it was mandatory, at that point, we could start telling people, you did not give us the name of the instrument. So we reject your data set until you give us the name of the instrument. And since they were mandated to do it to get their publication, they had an incentive to put the name of the instrument. So this is a bit of social engineering that has to come bit by bit. And the same thing happens with tissue, where in the beginning, all the tissues were unannotated. And then after we started policing, the unannotated went down and the annotation shot up. So that's metadata. It is a problem. And going back to the original authors, this is a paper that we recently published in Nature Communications about this problem, is whether the information is in the database. And you can see that lots of information is simply not even catered for in the database. But this is the data in the paper. This is the actual paper. And what you see is bars is not the presence, but the absence of the data. So in how many percent of the papers is the data not found. And you can see that quite a few of these very simple basic elements in mass spectrometry are not even in the papers. So very often this information, this metadata, is completely lost because it's written down in a lab notebook in the best case. But since the postdoc or the PhD student moved on, nobody even knows where the lab notebook is anymore. And so that is very, very dangerous, because if you want to successfully use data, at some point it becomes important to know more about the data. And if you let that information slide away, well, at some point the utility of your data reaches a hard limit. You cannot go beyond a certain limit. The data is still useful, it's just not as useful as it could be. And so that's why metadata is also a problem and will remain to be a problem for the foreseeable future. It's because people don't take responsibility for their data and they don't understand that the data is more important than the paper.
Thank you.
Sorry for the long answer, but they were good questions.
Another question. I suggest that maybe, Jérôme, if you can start setting up your slides while we are maybe taking a second question. For those we want to discuss in more details with Lennart, he can stay at the end a little bit longer, but so that we move on to the next presentation.
Hello, my name is Wiebke Drenkan. Metadata is one thing, but I think we all have the same experience. You ask 3 people to do the same experiment, they get different results. Because it's a question of purity, of procedure, of etc. So how do you make sure that the data that enters into the database is actually reliable, reproducible, etc.?
Yeah. So one of the things is, in the beginning, certainly you cannot. You simply cannot. You just have to accept the data as it is, and it's not reviewed. And so you have to take this into account in reanalyzing the data. So when you reanalyze the data, you're forced to do a quality control. So I didn't talk about that, but every data set that we ultimately use is quality controlled one way or the other by us. And why? But it's also a very hard thing to do in the repository, because data that is of sufficient quality to do the prediction of the fragmentation, for instance, is very different from data that is of sufficient quality to do the prediction of the tissue. And so each type of use, reuse, repurpose, reprocessing requires a different kind of quality control. And so that is very much a thing that you have to do yourself at some point. Now, what can help is if standardized means of quality control are at least provided, so that you don't have to recalculate quality metrics. And this is something that we're very heavily fighting for in the field right now, that every data set, both a posteriori, gets calculated quality control parameters, which are then publicly made available, but that in the future incoming data sets also present a certain list of predefined quality control metrics on their data that then can be used automatically to filter. And so I would say what I've learned is that one person's trash is another person's gold in this case. Some data sets that might not be good enough for my purpose might still be good enough for your purpose. And that is why making them available is probably the best thing to do, but then with the quality attributes annotated so that they're easy to filter, so that between you and me, we can each choose the data sets that we want without having to do too much recalculation. And for that, the field has to come up with some good quality measures. And the two things feed it into each other. As more people reuse data, they will become more knowledgeable about the kind of filters that they would like to apply. And so this is something that moves along. A good point here is, however, that a lot of these quality control metrics can be calculated. And that means that you can always do it retroactively for existing data. So it's a good point. It's something that needs to be taken into account, but it's not an unsurmountable challenge, I would say. Yeah.
Thank you. We have one last question for Lennart. Maybe if you can be as quick as possible so that we leave some time for Jérôme.
Pour Jérôme, je m'excuse, Jérôme.
Pas de problème, là.
I get a question about multiplication of storage of data,
Yeah.
and how to fight against climate global warming.
Okay. So interestingly, the storage of data is not that energy hungry in itself. Computation is very energy hungry. Storage is okay. Depends a bit on what kind of storage you have. If you have spinning disks, which is the classical way, they still require a bit of energy because the disks keep spinning. If you have SSDs, they require practically no energy. As like USB sticks, fundamentally. So if you unplug a USB stick, the memory stays there. It's only when you access it that you require a very small amount of energy. So the compute power is the expensive bit. So if you want to be climate efficient, and we should, there's 2 or 3 things you can do. First thing is limit the amount of computation. For instance, quality control metrics, calculate them once and store them with the data. Don't everybody calculate their own each time. That is already a huge impact. Second thing is, when you build models like we built machine learning models, don't build the fancy ones just because you can. Build the simplest model that works, that uses the least amount of CPU cycles. And then the last thing is, don't transfer the data back and forth 100 million times. If I need data sets, I store them locally, and then I use them until I'm done with them. I will not use them and then delete them and then download them again and then delete them, because that is wasteful. That also requires transfer. So the problem is not the storage of the data. Storage is practically free in terms of energy. It's all the other stuff, and there you can be efficient with a little bit of care.
Thank you. So we switch to the second part. It will be much shorter, like a few minutes, just to introduce you with local tools. So Jérôme, the floor is yours.
Yes, thank you very much to give me the opportunity to present the ADELE Helpdesk. ADELE Helpdesk is an Alsatian ADELE Donnée, that's a group of people helping you to manage your data, but not only to manage your data, but also to share your data. It's, of course, in a relationship with the previous talk of Lennart. Where is coming the ADELE Donnée from is included inside the national ecosystem, but above the research data group project. I hope that most of you are aware of this project. The aim of this project is to provide a national repository for all the data that don't have dedicated scientific domain-specific repository, as we could see before, like PRIDE. Also, you can put all your data that can be shared on this repository. Of course, it's mostly done for data that are clean enough to be published. This is not done for raw data. There is another solution for this kind of data. So who is involved inside the ADELE Helpdesk? Of course, the University of Strasbourg, CNRS. CNRS has two institutes that are involved in through the CNRS, the MICHA for humanities at the central campus, and IPHC through the science platform that is on the Cronenbourg campus. So I will be very short on this presentation. I am still available either for during the meal today or on other day if we want to discuss more about the ADELE Helpdesk. On other institutes that are involved is INSA, so the engineering school, Université of Alsace, and of course the BNU, because it's very important that librarians are involved in the management of data on software on publication, of course. So they have a very high value for helping scientists and IT people to share the data. So I will go over this slide quick because it takes over the same information than before. The scope of ADELE Helpdesk is to provide the support for sharing, managing your data for most of the actors in Alsace, either in Strasbourg, Colmar, for example, in Rai in Colmar, or in Mulhouse with Université of Haute-Alsace. The main aims of the ADELE Helpdesk is to provide support and guide researchers through the data lifecycle. So the overall lifecycle of data, starting at the project planning, when you have to create or get your data, manage your data collection, how to store, where to store, how can you do long-term archiving, how to share your data, what are the main issue you can have when you open the data, you have to choose the license, and so on, and how can I reuse the data with most of questions for coming through legal issues. There was one before that was very interesting about what are the licenses that can apply or when I am using data generated from IA, for example. So that's shortly some solution examples. For example, the ADELE Helpdesk can help you to put database scoring in place through the use of API when you want to automate your collection of data. There is the help of something is very important. And Lennart spoke about this before, is the metadata schema recommendation, how to automatize the generation of the metadata. Of course, we want to make it as simple as possible for scientists to share the data. A question that is coming even often is long-term archiving of data. Either through a simple backup, but for long term with the use of the tools in CNRS. CNRS is a national data center in Montpellier, but that has a goal to have a lot of services about long-term archiving for all the Ministry of Research in France. For long-term archivists, so people from BNU will help you, and the data protection officer can also help you both on the best practices like LSPEX. On the ADELE Helpdesk is here for helping you, of course, on this. Data processing, storage, there is a lot of actors on the place, so don't hesitate to come contact us if we have any issues. Concerning the data and software reuse, we can help you to set up DMPs. That's very important because most of the projects today need a DMP, either RNR, European Hash 2020 projects, and so on. We can also help you on choosing a license to open your data, to open your source code, and give you some hints to our best practices to when you want to open your code. That's very important to be to make reproducible research. So that's all. I was very quick. If you have any question, you can contact us on this through this email. I will stay after the talk. So if you want to have some exchange about this talk, I am available.
Thank you. So really the idea is that you know who to contact if you have more in-depth questions about how to share your data. So for those of you who want to discuss more, we can stay here. We will also, like for lunch, anyone who wants to join at the course with us is welcome. So with that we can continue discussing those things. And if there is like any urgent question for Jérôme. Yeah, Noémie.
So you mentioned SIN. You are in charge of SIN platform. Can you just in two sentences explain what is SIN?
Yes, SIN is a compute and data platform, so data IPHC, but open to all the researchers and ITs, people from the university, but at the larger scale, at the France and the European scale. We are providing HTC resources and cloud resources for computing data. And we are providing expertises to help you to manage your data. And we are also providing data management solution if you want to store data and analyze your data. And we are, of course, coordinated with César group from the DNUM, that's the Direction du numérique at the University of Strasbourg, to give you global access to computing power and other services around data.
Thank you very much. A painful question, maybe. We are one of the last unités propres du CNRS. How do you make any difference between CNRS people/labs and university labs in terms of like efficiency of treatment, or like what, like the service that you offer for them, or this is for you, everybody the same?
It's for the same. As you see, CNRS is a partner of the ADELE Helpdesk. There is an official letter from the CNRS that gives us the freedom to help all the CNRS people. So no difference for us.
Any other question? Just one remark, there are lots of guides that have been brought here so that you can help yourself on many different questions on open science, open data, things about publications, copyrights, like things like that. So feel free to take whatever you want. So let's thank again Lennart and Jérôme